Good morning, good afternoon to those joining us from different parts of the world. It is a great pleasure to welcome you to another session of the UNAM-UK Student Seminar Series, an initiative that is designed to highlight the academic and scientific work of Mexican students in UK and beyond. We are delighted to have joined us in this virtual space where you continue to build bridges between academic communities from an international, interdisciplinary, and collaborative perspective. To begin today's session, I am pleased to introduce to Carlos Neri-Cruz, who will present in the talk entitled Nanoparticles Functionalized with Polymer Brushes for Gene Delivery. In this talk, Carlos will share with us how polymer brush-functionalized nanoparticles can be designed to control the delivery of genetic materials in biomedical and diagnostic applications, with a focus on physicochemical tuning and emerging possibilities in the diagnostics. This session is being streamed live on YouTube, and we invite you to share your question in the chat through this session. We will reserve some time at the end to address as many of them as possible. Before we begin, allow me to very briefly introduce our speaker. Carlos Neri-Cruz is a PhD candidate in Biomedical Materials at Queen Mary University of London, where he investigates how polymer brushes macromolecular structure composed of densely tetratered polymer chains can be designed to improve RNA delivery. His work aims to contribute to the development of safer and more effective genetic therapies. Carlos holds a Bachelor in Science Chemical Engineering, specifically in food science and technology, and previously he works in research and development at Grupo Bimbo and Enterprise in Mexico. His research is supported by CONACYT and supervised by Professor Julien Gautreau. He has co-authored several scientific articles and regularly presents his work at international conferences. Then, Carlos, the microphone is yours.
Great. Well, thanks so much for the kind presentation, Dr. Del Rio, and thanks a lot for having me. I'm very happy to be here to talk about what we do in our lab. So before I start my talk, I would like to briefly introduce the team. So we work in the Gautreau Lab by Interfaces for Life Sciences. Our principal investigator is Professor Julien Gautreau, and here we have a picture of all of us. Maybe I'm missing some of the faces, but essentially this is our group. And well, let's get started. So today I'm going to talk about a topic that is very interesting for me, and I really like it. And is about a specific class of nanomaterials that we call them polymer brushes, and I'll explain later why we call them like that. And these polymers are very interesting because we have observed that they can complex RNA or DNA to enable the delivery into cells for several applications, including gene therapy, but also for vaccine development. So this is kind of the outlines. I'm going to talk about first a bit of gene delivery therapies, then how we can structure these polymers, and finally how these polymer brush RNA complexes are formed and what are the main strategies we are trying to develop in our lab. So gene delivery. So why do we want to deliver nucleic acids? As I mentioned before, the delivery of nucleic acids is, I think, right now quite important, and perhaps it's becoming more and more popular, especially after the COVID-19 pandemic, where we observed an increased interest in the development of vaccines. For example, the mRNA vaccines from Pfizer and Moderna. but it's not limited to vaccines. Also, there are applications in gene silencing and gene editing. I think it's quite interesting that if a gene in a patient is malfunctioning, we can essentially turn it off or knocking down this gene so that we can find a way to cure or treat diseases. So essentially, this is what gene delivery is. And normally, to deliver nucleic acids, we use vectors, which are essentially vehicles where these nucleic acids are being incorporated for further delivery. Although also we can deliver nucleic acids in a naked way or naked format. However, I think we should think about this kind of as a whole, because yes, naked nucleotides or nucleic acids, as long as they are specifically designed and modified, they can survive the physiological conditions where there are a lot of molecules, for example, enzymes that can degrade them, but they can still survive as long as they are nicely designed. However, this can be limited, so that's why we need a vehicle, a way to protect these payloads. So we call them vectors, and that's essentially what I'm gonna be talking about. So gene delivery, gene therapy is, yes, as I said, is becoming quite increasingly popular. However, we are, I think, in very early stages still. There is only a few number of drugs that are commercially available, and most of them are for the treatment of genetic diseases only. And now, well, also some vaccines just recently. However, we still see a lot of challenges, especially in terms of safety. Many of the issues in translating these drugs into clinic are associated to the toxicity either from the vector or from the cargo or RNA, DNA itself. So this is something that we need to address, and this is one of the very important reasons why we don't have too many drugs currently available. And together with that, the exorbitant cost of some of the treatments. For example, a very interesting drug that is used for the treatment of spinal muscular atrophy. It can cost up to 2 million almost, or 2 million pounds per dose. So imagine that that for a patient is a lot. That is especially for the government who is actually funding this is quite a lot, and it's not only one dose. It needs to be several doses. So that's one of the challenges. And perhaps the most important I consider is the knowledge, because it's true that we know a lot about gene therapy and vectors, but we still don't know many things, many aspects associated to the delivery. The delivery of nucleic acids is quite complex, from the formation of these vehicles until the release, and once these nucleic acids reach the target cells or the target organs. So I think we still need to understand what is happening, and this brings the idea that we need vectors that are very highly defined and highly controllable to be able to really study systematically all these complex aspects. So one of the candidates to study this are polymers. And why we like polymers? Well, because polymers can be designed and can be synthesized in a relatively easy way, with high definition in terms of chemistry, in terms of architecture as well. So that's why we like them. There are other vehicles, lipid particles, for example, but in our case, we like polymers and we focus on that. To begin with, before I move to particles functionalized with polymers, I would like to introduce the traditional approach, which consists of essentially polymer chains that are positively charged. They are polycations, and these polycations, because they are positively charged, they can assemble, they can capture and self-assemble with a RNA or DNA that, if you remember, they are negatively charged, in forming these complexes. However, gene delivery nanomaterials that are based on self-assembly, they are challenging to control. Why? Because most of them are, actually all of them are kinetically and thermodynamically driven, driven by the formation of this. So that means that any change in the temperature or any change in the conditions, especially in the physiological context, where we have a lot of things happening, it can impact on the complexation and can impact the shape and the behavior of these complexes. So that's a point that is tricky, that is challenging with free polymers. And also, if we want to post-modify or further functionalize these systems, we have the risk that the complex or the assembly simply will just dissolve, so we can compromise the vector itself. So that's not great. Though, of course, there are several approaches with these traditional methods that are getting more and more control. However, can be challenging. So what if, instead of using polymers that are free and that assemble freely, why we use polymers that are immobilized on surfaces, on nanoparticles, for example? That's quite interesting, and we have several approaches actually. So just to give you a brief idea of that, we can, for example, physically absorb polymers onto particles, something like this. This is perhaps the simplest way to do it. However, I'm not a big fan, because still you can see that it's just a physical absorption. So the polymer itself, it still displays a bit of freedom to adopt random conformations. This can lead to a bit of entanglement of the chains. So the control over the architecture of the polymer is not great. And also, something that we need to bear in mind is that this is physical absorption, which means that perhaps, well, not perhaps, actually, there are reports that many systems experience detachment of the polymer chains after, for example, after washing or open functionalization. So that's one of the weaknesses of this approach. But instead, we can rely on the covalent grafting of polymer chains. So the polymer grafting of polymer chains can be divided by two. So either the grafting to approach or the grafting from. So when we say grafting to, it means that we can generate polymer chains ex situ. We can just design them and then modify them in the way we want, and then we can couple these polymer chains on particles, providing, of course, that these chains are active. They have some moieties that can react with the cores. So that's one approach. And the second one, the one that we work mostly in our lab, is the grafting from approach. This approach is very interesting because essentially the polymers are generated from the core. So you have a core, a particle that already contains some molecules, and I'll talk about that later, but some molecules that can initiate the growth of the chains. And because we generate a lot of chains over the surface, we generate highly dense polymers that because of the repulsion, the interchange repulsion, they adopt this brush-like conformation. So that's why we call them polymer brushes. And let's, for some reason, it's not changing. In a sec. Oh, yeah. Okay. And here I would like to make a quick pause on the topic, because I think it's important to explain more in detail how we generate these polymers. Because in the previous slide, we saw that, I don't know if you noticed that there was an acronym, CRP, which means control radical polymerization. So we can generate polymer brushes very well defined with these approaches. And one of the techniques that is highly controlled is the atom transfer radical polymerization. The acronym is ATRP. And it's quite interesting because, before it was developed by Krzysztof Matiłaszewski's group in 1995, so some years ago. And essentially, it relies on the equilibrium between dormant species and the activated species, active species. So if you look at the formula, the chemical equation, actually, it's quite simple to understand. We have from one side, I don't know how I cannot move my well, it's fine. So essentially, from the left, we can see macromolecular species. This can be anything. For example, it can be a monomer, or it can be a initiator molecule that contains a halide, and then we have a metal and a ligand. So we have this system. Halides are very good living agents, living groups. So in the presence of a metal that is complex with a ligand, this halide essentially leaves so that we generate a radical. So it's this asterisk on top of the macromolecular species, now on the right. And this is now a radical that is active and that will enable the coupling of a monomer. So we can couple a monomer. But then the interesting part here is that you can see that this reaction is reversible. So we add one monomer, but then the reaction gets deactivated again by the same halide. So we deactivate, we stopped, but then we come back, and then we add another monomer. We stop, and we come back, we add another monomer. So this is great, because then we can virtually add monomers one by one, and that's what is providing with control on the growth of the polymer chains. Before these approaches like ATRP, there was just free radical polymerization, where the propagation of the radicals was uncontrolled, but now we can control it. Therefore, we can really deliver polymers with very low polydispersities, so well defined, with the polymer chains mostly around the same molecular weight. And also we can generate a lot of different architectures. So that was a very nice invention back in 1995. And this is the one that we use in our lab to grow our polymers. Okay, well, and so now that we know what polymer brushes are, I would like just to emphasize why we like them. We like them because we can generate polymers at very high densities, super high. And this will gain relevance after. I'll explain why high grafting densities are great. But that's one of the characteristics. The second one is that they are core independent. It means that independently of the core, we can characterize the polymer chains, and the core is just a vehicle or a way to immobilize. Of course, the core can also provide with other diagnostics applications or features. For example, we can use metallic particles to guide the targeting of these drugs, or I mean, it can be many things, but I'll explain that later. In terms of the polymer, we can control the thickness, right? Imagine that you have your system, you have your particles or your substrate. You can generate polymers, and you can decide when to stop the reaction. in order to get the thickness you want. Now you can end up with very large, very thick brushes or thin brushes, and also you can tune the grafting density and so many things. So that's why we like that. A few examples of architectural designs are shown in this slide. I was telling you before that we can tune the grafting density. It is very interesting because, for example, we can couple to 100% of these alkyl halide initiating molecules that I explained before in the ATRP process. And essentially, all the particles will be covered by polymers, right? But what we can also do is we can, rather than using 100% of silent, 100% of initiator molecules, we can prepare a mixture of initiator and another molecule, a dummy molecule, a molecule that is similar and that will attach to the core, but that is not reactive, it's not active for ATRP, which means that we can then generate a lower grafting densities where the separation between changes is sparser, no? Or it's larger. So these polymers, strictly speaking, they may not be considered polymer brushes anymore because they are now adopting a mushroom-like conformation. So rather than being a true brush, but still in the field, we call them low-density polymer brushes or sparse polymer brushes. Yeah, but they are interesting because then if you have a higher separation or larger separation, then, for example, if you want to capture a big molecule, then you have space. While here you have space, and here this is for the high density systems. We have seen actually that these are great to complex small molecules because of the very high density, which I explained in a bit. We can also design block polymers. For example, that's very easy because you generate a first polymer chemistry, but then you can reinitiate the ATRP reaction to generate a second block from a different chemistry. Some authors have also functionalized particles with more than one polymer chemistry. You can use, for example, 2 or 3, by simply using different functionalities, initiating functionalities. So we can end up with something that is mixed polymer brushes. So we have plenty of field to play, which is great. And apart from the nanostructuring of polymer brushes, we can also functionalize. We can modify a bit the chains, either the side chains or the end chains, with molecules that can enable the coupling of, for example, a peptide, which is quite relevant in the field of gene delivery, because if we want to target certain cells to reach certain cells or certain organs or tissues, we can couple a peptide that will guide the vector to only reach this kind of, or that a certain kind of cells. Here we have an example, very classic, where we have a polymer chain that is functionalized with an azide group, and then we have a peptide, RGD peptide, that is contains an alkyne, and simply click chemistry reaction azide, alkane azide, we can couple that. So they are quite versatile in terms of biofunctionalization. And well, because of these functionalities and these characteristics, our lab has been very interested in developing polycations to deliver small nucleic acids, and specifically siRNA. siRNA are small RNAs that can silence the expression of a gene. So, for example, here we have one of the gold standard chemistry that we use in our lab. This is polydimima. It's a polycation. It has a tertiary amine here that is can be positively charged at physiological pH below. And essentially, you can complex RNA and deliver this RNA to cells, enabling the silencing. If you see in this panel here, we can see the silencing of GFP actin by using a gold standard product. This is like Vectron, it's a lipid-based particle, and a polymer brush. And you can see that the reduction in the intensity is due to the reduction in the expression of the protein. So we can nicely silence proteins with these systems in comparable levels as for lipofectamine, which is commercially available. And I think the most interesting aspect here is that this silencing is dependent on the architecture of the polymer, because we have found that high density polymer brushes and thicker brushes are actually better. If you see this graph, this is a graph here showing the knocking down efficiency in these cells. And we can see that this is the efficiency for lipofectamine. This is for thick brushes around 30 nanometers. But then as you move to lower thicknesses, then the efficiency is decreased. So we can actually control the efficiency just by controlling the thickness of the brush. And perhaps more intriguing is the role of the grafting density, so the distance between chains. So here 100%, 10 nanometers, 100% means that we have a highly dense polymer brush system compared to 5% or 10%. You can see that there is a big difference, right? So 100% highly dense polymer brushes are better to disperse brushes for the delivery of small nucleic acids. And well, why? We didn't know much. One of the hypotheses was that perhaps this highly dense and highly crowded environment of chains enables the infiltration of small nucleic acids. However, just recently we confirmed that in this study that we published this year. And I'm going to move now on how these nucleic acids interact with polymer brushes, what is happening really, and if this is actually what accounts for the highly dense, for the high knockdown that the group has observed before. So here we have some plasmon resonance essays, and I know we have here a lot of things. Let's go one by one. So before I explain the graphs, surface plasmon resonance is basically a technique where we measure the plasmon resonance that is the product of the interaction between a molecule with a sensor. And this sensor is essentially just a glass slide that is, or glass chip that is coated with gold. The gold is the one who is in charge of producing this plasmon resonance, and it's coated with polymer brushes. I showed you before that we use these polymers grafted from nanoparticles, but because this polymer, the generation of these polymers is core independent, we can generate these polymers from any surface. So that's why also we like them. It's very convenient because then we can study separately the polymer RNA interactions with these techniques, with interfacial techniques such as surface plasmon resonance. So what we are observing here is the injection of different nucleic acid material. We have mRNA, large, 1,000 bases, and we have small nucleic acids. We have single-stranded RNA and double-stranded RNA. And essentially, we just inject a solution on top of the polymer coatings, and then at different concentrations, and then we can see an increase in the binding. So for mRNA, we observe a binding of around 2 micrograms per centimeter square, but with large molecules, right? But you can see a big difference when we try to absorb small nucleic acids, polymer brushes, we end up with 3 to 4 micrograms per centimeter square, so double the amount. So you can see it here in summary, right? So RNA, small nucleic acids are better absorbed. And interestingly, if we look at DNA, plasmid DNA, which is huge, is circular, is a huge molecule compared to single-stranded of 20 base pairs, right? Here we have 5,000 base pairs, which is a lot. Then they're poorly absorbed, similarly to mRNA, right? But interestingly, also for double-stranded DNA, which is small, but then the chemistry is different. So we found that first, there is a strong impact of the size of the molecule that we want to absorb, but also there is a preferential binding for RNA over DNA. So that's interesting, and that's an interesting point to consider in the design of nanomaterials for gene delivery. With this data, we used a validated kinetic model. And this model is quite interesting because it looks at the interface between polymers, between the polymer brushes, so at the top, and the media. And from this analysis, we can extract some constants, for example, affinity constants that tell us a lot of things. So in this graph, you can see if we only focused on the blue bars that are the affinity constants, we can observe that larger molecules, mRNA and plasmid DNA, they display very high affinity constants, which is a bit contradictory, right? Because we observed before that actually mRNA and plasmid DNA are the molecules that are being absorbed in a lesser extent. So a bit contradictory, right? Or maybe not. Maybe what we were wondering, perhaps the high binding of small nucleic acids is due to the fact that indeed these nucleic acids, small nucleic acids, should be infiltrating through the polymer brush, and actually they don't prefer to stay at the upper layers, right? So they don't like the upper layers because they infiltrate. So of course there is an initial absorption at the upper layers, but there is then infiltration, and that's what was kind of a hint in this study. But to confirm this, we did some neutron reflectometry experiments, and before going to that, I quickly need to connect my computer because I forgot it. So give me just 1 sec and I'll be back. Okay, that was quick. So, uh, so then we try to resolve the architecture of these complexes by neutron reflectometry. And I would like to thank Dr. Ali Sarbach, a professor as well in Queen Mary University of London, and also to my supervisor, Julian Gautreaux, that they did a really good job at resolving these and fitting all this data. And again, we have a lot of things, but I would like you to focus on these panels. These are SLD graphs, and what we can see is essentially the scattering length density changes as we move from the very bottom to the upper layers, right? So from 0 to 80 nanometers, 100, right? Because these polymers, when they are swollen, they reach something around 100 nanometers in thickness. So if you observe here for mRNA and for plasmid DNA, the upper layers, we essentially don't see any shift in the SLD, independently of the concentration, right? So we have zero, like nothing, just PBS is the blue lines, and then 3 and 10 is green and red, right? So we don't see any change in there, right? For mRNA and plasmid DNA, we don't see any change here. But if we move a bit more to the left, Here we can see a change. It's small, but it's very important. But then if we move to single-stranded and double-stranded RNA, we don't see anything, like no change at all in the very up layers, but also in this region, we don't observe anything. So it means that in these large molecules, they are actually absorbing at the top, not top, but still top layers of the brush, while single-stranded and double-stranded RNA, small nucleic acids, they prefer to infiltrate. So this schematic illustration try to illustrate, right, what is happening. No? So it was very interesting work, and I think we got a lot of insights on how this complexation is happening. However, so yes, we know that the high grafting density allows for infiltration, stabilization, and protection. Yes, but apparently it's not enough. And it's not enough because upon entry in the blood or in the cytosol, there are a lot of molecules, proteins, carbohydrates, RNA and DNA and enzymes, a lot of things have around that they can compete to bind the brush, triggering the release of RNA. So we call it a competitive desorption. So it really doesn't matter if you have a high density brush, uh, because this protection is limited, right? So this is very important because this is a challenge if we want to translate into clinic. We need to engineer polymers that are that provide with enhanced protection to avoid these interactions. So what can we do? Well, well, I'm not presenting all the work I'm doing. This is an unpublished paper that hopefully will be published soon. But essentially, I would like just to give you a general idea of what we do, right? So here we have a particle that is coated with a block of polymer. We use block of polymers, and we have one chemistry that is the chemistry that have been discussed so far, polyvinyl. But then on top, we have generated a second chemistry. This is a polymer that is thermoresponsive. It's quite interesting because at room temperature, 20 Celsius degrees, it's completely open. It's essentially extended in conformation. But then at 37, at physiological temperature, collapses because there's a change in the hydrophobicity that makes it collapse. So then we are proposing that when it's open, it can allow the loading of RNA, but then as soon as it reaches the body or the cells, it collapses, it chills the system, and then it's chilling the interaction with these proteins. It's essentially slowing down the competitive desorption. So I just can tell that we have observed very nice results, and hopefully I can share this paper soon. But well, essentially this is the strategy. So I think that's all from my side. So I'll be very happy to receive any question you may have.
Thank you very much, Carlos. We have some questions for the public. Sergio
Okay.
Ramos asks, could these brushes be adapted for reversible gene silencing? Or are they designed for permanent knockdown effects only?
That's a very good question, actually. I mean, you can do both. You can certainly do both. Uh, However, in the gene therapy field, uh, the research is more focused on knocking down, so not complete knockdown, knock off, right? So just a partial silencing because it's safer. If the RNA, even if it's, I mean, it's very specific, to a certain sequence, but still can have some off-target effects. So it's safer if it only knocks down rather than it knocks off. I don't know
Okay,
if you can. Yep.
and you can tune the response if
The response
in the
to
same process. Can I imagine only there even one thing you can really do reversible as you want?
okay, I think yes. So one of the strategies is and one of the actually one is very important aspect. Ideally, we should engineer systems that allows the sustained release of RNA and prolonged, meaning that at the at day one or day two, day three after the injection of these therapies, you see knockdown, but then you can engineer this system so that the effect is not that profound, because it may be that perhaps it's very strong for a patient. So you can, for example, use these polymers to slow down the release of RNA. So you can tune that part, and also you can have for example, chains that can degrade under certain conditions in the cell, maybe by in the presence of reducing agents, so that you can expect the release of RNA not immediately, but after maybe 3 or 7 days after you inject it. So you can certainly tune the release and the final effect, just looking at the polymer chemistry and the architecture.
Great, sounds wonderful. We have here another question. I may Castel ask you, could this technology be adapted for targeting delivering to specific organs or cell types, such as brain or tumor tissues?
Yeah, definitely in the group, we are also work on that, especially one of my colleagues, Chris. So yes, he's developing systems where so essentially similar polymer brush chemistries, but then you can couple a peptide that will recognize specifically the surface receptors of certain type of cells, and actually he's working with glioblastoma cells. So yeah, definitely we can do that, and it's challenging, yes, but the chemistry part is actually quite doable, feasible. Essentially you can use, there's a whole toolbox of click chemistries and coupling chemistries to use, and yeah, it's possible.
Okay, maybe I couldn't understand very well, but can you briefly explain the role of protein corona formation in nanoparticle behavior?
Yeah, of course. So essentially, uh, in the cells in the blood, there are a lot of macromolecules, but specifically, mostly proteins. They they absorb easily because they are charged. They absorb easily to these polymer coatings. And other vectors is not exclusively on these systems, but on particles, specifically because they are negatively charged. They can bind cationic polymers, and essentially they coat the systems, and this coating that is competing to bind the brush, so therefore releasing the RNA. So it's an exchange process, and we don't want that. We want to avoid it, actually, because if if the particle or reaches the cell or the blood and immediately after releases the RNA in a burst way, then that's something that is preventing us from delivering long-term and prolonged or sustained release. So yeah, that's the role of the corona.
Okay, thank you.
Yeah.
Another question, how do you evaluate cytotoxicity or biocompatibility in your experiment?
So what I normally do is so we normally start with simple live death essays. We haven't really, so for polydinema, for example, which is the gold standard in the group, we get a relatively high viabilities, something around 90% viability, which is quite good. So these polymers, they are not really that toxic in that sense, though we have all the chemistries that we have tested and they are very toxic, but we have identified some by live death, uh, but that's the first stage. Uh, what we are doing as well now, we are looking at the interactions of these polymer vectors with proteins by proteomics analysis. So we now know some off-target effects that are happening upon interaction with the cells. And yeah, we have a set of different chemistries. Some of them, they have a lot of off-target effects that are as essentially toxicity associated, but there are others that are still quite good, though we haven't reached really like preclinical stages, so we are still in early stages on this, but that would be the one way that two ways that we are testing the cytotoxicity.
Okay, and what's about the real application? What are the main challenge you face when translating this research into clinical applications?
Yeah, I think coming back to the previous question, I think is the toxicity associated to the vectors, the polymers, but also to the RNA or DNA. I think that's the main challenge because most of the candidates, polymer based, that are in the clinical stage now, most of them they have failed and they're failing because of toxicity issues. So that's the main challenge. Uh, because it's not surprising because cationic polymers, they're cations. They can interact with cells. They can disrupt the membranes. They can interact with organelles that we don't want to. So that's why now the field is moving to generate block polymers, for example, where the second block is kind of masking this cationic potential, so that you are limiting the interactions. So the less the interactions that the vector can have with the cells and the components, the better, I would say.
Okay, thank you, Carlos. And was a last question. You told us that you are almost finishing your PhD. Then the obligate question is, what are your next steps in your research after this PhD project?
Okay, well, that's a nice question. So yes, I'm almost done. I'm submitting my thesis in December. So I'm in love with this field, to be honest. So gene therapy and essentially the engineering of delivery vectors. I think I've found one of my another like passion in my life. So I would really like to continue developing uh, my scientific career in this field. So the next step, it would be to become a research assistant, to find a postdoc position. So I'm actively looking for, uh, but I would like to stick to this field. Of course, I'm open to other kind of materials, though I think polymers are great. So let's see.
Perfect. Well, thank you very much, Carlos. Thank you very much for this very nice presentation. And um, I'm sure you will find uh, ways to follow this research.
Okay,
And
thank you. Thank you so much.
thanks to the audience, and please be attent of our networks, and for we expect the next 2 weeks the following seminar. Thank you very much, all you.
Listo. Muchas gracias, Carlos. Muchas gracias,
Listo.
doctor.
No, muchas gracias a ustedes.